Okay, good morning. This is week 8. So we are going to talk about some kernel functions and the kernel concept, which machine learning people used a lot especially in the classification application area. So I want to also show you this someone in LinkedIn post visualization for the neural network. We learned the perceptrons, and then each of this is a perceptron. Then the input will be aggregate and pass on into another layer. So when you have a lot, and then you make a network. So this is like a nice visualization for what's happening in the neural network. Okay, all the links are the information passed on, and the sake of this is how big the parameter has been tuned into, and then depends on your output. This parameter can change, and the weight of the linkers will be changed. Okay. By the way, the homework was really good, and I told you it was very easy, right? You haven't lost any blood so far. So this week we are going to have a new homework. Will be very different. Will be this never happened before, so this is called VIBS coding. Have you heard about this? So we are going to use ChatGPT or language model to help you to complete your homework instead of your own. So what we are going to do this is you will have a paper. You pick a paper and then ask me whether this is a methodology paper good enough. I will say yes. Then you go ahead and upload the paper to ChatGPT or other language model you have, and see. Please follow the paper and the methodology description, and implement this method in MATLAB. See whether you can successfully train or promote the language model to generate the code for you, and then you put into MATLAB to test. See whether the input and output are expected as you hoped. Why we are doing this? Because I don't know if you have GitHub. If you have GitHub, so this is a place where programs will upload their code. Sometimes it's for share, sometimes it's just for keep your own record. I don't know how many of you have this GitHub. Oh, one of you, two. So this is basically for programmers. If you have done some coding, then you have this activity. It's called contributions. Then, for example, that day in July this year, I upload and download 11 times. So I have a record. So this is basically show you activity of your coding. So if you look at my coding, so I basically do this every day, especially except some holidays. Maybe I don't have access to the internet. From this activity, I'm a very experienced programmer. So I use MATLAB a lot. I can do R and Python as well, so mixed. However, last month, or just last week, I realized this web coding. So AI is already is more powerful than myself. So it's a tipping point, which everything changed. So you don't have to rely on your former experience of coding. you can ask language model to do it for you. In Texas A&M, we have, I don't know if you have this access to this chat.tamu.ai. Do you have this access? I pasted it here. I don't know. I have to. Let me point out this. Do you have TMU open web UI? Because if you don't have this, may have found different ways to do this homework. Just let me know. Do you, as a student, do you have an account in here? Yes or no? Yes. Okay. As a student, you have. Okay. That's good. Then we can do this. Okay, come back to today's topic. So I will start by revisit some key concept, some like dot product, etc., etc. Then we'll introduce a support vector machine. Okay, there one of the options in your classification learner. Remember last week in the computer session, in MATLAB, their classification learner, one of the options is to use support vector machine, SVM, to do the classification. So beside that, you have KNN, okay? K nearest neighbor. You have LDA. You have neural network. But the support vector machine is one of these class, a group of the algorithm which build around this idea. And I need to introduce this to you because it's a classification lecture. Then there's a feature mapping function or kernel. The concept is useful. So after these two, and then we will discuss single cell, cell-cell interaction or cell-cell communication analysis, how this analysis will be done, and what's the principle behind this. Okay? So we have done this many times, dot product. And all you have seen so far is all about the dot product, including the neural network I just showed you. Okay, so very complicated. Even everything behind this capacity is rooted from the transformer. Transformer is one of the neural network framework, okay? So everything boiled down at the end is dot product. What you have A is 1, 2, 3, and the B 4, 5, 6. And to do dot product between these two, you need to put this A horizontal, okay? 1, 2, 3, and put this 4, 5, 6 vertically. So this follow the rule of matrix multiplication. And then you get this 1, 1 because you are computing the multiplication between every elements, then sum them together. So how to do this? It's 1 times 4 plus 2 times 5 plus 3 times 6. Then you get a single number at the end. So for one vector and another, the dot product is the same as you do this element-based. So you do 1 times 4, second 2 times 5. So dot product is element-based multiplication and then sum them together. Nothing. So that's basically the concept of this is you can consider this dot product as the magnitude of the first vector times the magnitude of the second vector and then times their angle and the cosine of the angle. Homework, you know, if you consider this as your axis, then you project your vector onto it, and then you can quickly compute this projection, the shadow of this lens. Especially if you have a B normalized, if you have a B divided by its norm, by its magnitude, so this become a normalized B. And then you can just multiplicate. So B is normalized here, and you can just multiplicate with A vector multiplication. Then dot product, then you can get the length of the projection. Okay. We also studied LDA as a comparison with the PCA. PCA, you don't consider the classification. You don't have this blue circle and the red dots. You will treat them as a single group. Then you will learn this black line will be the axis which help you once you project your data onto this black line, you will have a most variability or most dispersed points. However, the purpose of LDA is to separate red dots from the blue circles. The solution will not be display the overall magnitude, the dispersion, but is to find this angle to look at the projection of the points by separating them as clear as possible. Okay, so if you do LDA, you will find the first principle, the first axis should be this green line. Well, once these points are projected on this green line, and the center of these two class of the points will be most separable. Okay, what most separable means the projected center will be more, the distance between the center will be most maximized. Well, sometimes LDA will be compared side by side with SVM. There are different, they all this linear algebra based. Okay, there's no neural network or other framework. It's just found the separation, found the way rotate the data is from LDA. Will SVM is slightly different. We will get into there very quickly. So okay. So before that, let's see. Okay, how can we use this very simple linear algebra operation to as a classifier, okay, to group the points. So in this example, we have some red dots right across, some here. And then we have some green circle over there. They are in this vector space, okay? So what we can do to build a very simplified linear algebra or dot product based classifier, so dot product based. Classifier. We first look at the center of this, so this vector, C1. C plus, okay? We found the center of this. Those are the known solutions. We know that they are training data. So this is vector 1, so called C plus, and then we have center here called C circle. So these are two vectors. Then we can compute a difference between them. So this is called a W, okay? So this is very easy to compute. W is, we can have a C minus, so difference between two vectors, so easy to compute. We can also get the center of this easily. So this is called a C. That's called C. How to compute the C? So you just this point plus this point divided by 2, okay? C equals divided by 2. Very simple operation, just vector-based plus, minus, take average. Okay, so then we are ready to do things. So the new input data will be, so we have a new data called, okay, so this come in, and this is called X in there. Our job is see, okay, whether this new coming in data is belong to red group or green group. So this, how this classifier work, it's just have a, you just compute the difference between X, the new data, to the center C. So basically you compute a vector here. You compute a vector here by using C minus X. Very easy to compute. Or X minus C, so it's all the same. So after that, we will have a dot product between this vector and this vector. So this dot product will tell us the angle, okay? will tell us the theta. So especially if you normalize by their norm, x minus c. So we are using this theta. If it is a positive, then belongs to this. If negative, belongs to that. Okay? So theoretically, it's basically you have a orthogonal vector which orthogonal to this w, and then depends on the angle. If the angle is 0, then it belongs to in the middle of this. If positive, belongs to this, negative belongs to that. Okay? So you can see this very small device and the dot product based classifier using the very simplified dot product to make this direction based classification algorithm. Okay? So that's the idea. So we want to use the simplest operation as simple as possible to achieve those designed goals. Our goal here is just whether this new point centered point towards the center of this cluster or this cluster. By the way, this example is in the a textbook, not a textbook, a book called The Kernel Machine, as one of the earliest simple example of this. We'll support vector machines more complicated than this, but still the idea is to use the most simplified linear algebra operations to achieve the goal as quick as possible. Okay, you may think it's not so essential if you are doing some of these classifications, small scales, but if you have a larger scale data set, we need to find a way. This is a way very quick, right? So compute center and several operation of the vector operations. So this machine idea, as I mentioned in the first class, it's when this neural network start, then I have a machine Turing, and remember in 1963, okay? This idea about support vector networks has been proposed. Vipnicking, okay, by Vipnicking, and then he further developed this in 1995. Finally, there's another paper by this Vipnicking, and so he has this illustration of what is support vector network. Okay, the problem is for classification. So we have this cross, dots, and the circle. So it's the same idea as this, okay? You have training data. So the support vector, the algorithm is try to find a divider. So this is a straight line which run cross in the middle of these two groups. And what is support vectors? Support vectors are data, are points, right? So support vector is this gray one. They are close to this. They are nearest data points, close to the divider line, the separation plan or separation line. So those are called support vectors. It supports the classification, to support the separation of these two classes. This is a supervised learning, so you need to have training data. You need to have this red, green, you know the separation, you know, okay, they are located differently in the space. So to separate these two classes, you can have this separation, this divider, or this line, straight line, either this one, or this one, or this one. There are many different solutions. They all work, and they all 100% or successfully separate red circle from this green plus. But which one is the best solution? So we need to make our classification algorithm, or this classifier, more robust and to the data. Okay, if you have noise, you have a inaccurate measurement. For example, if this point's moved, okay, slightly wiggled around this, and so which separation, which this line to separate them are the optimal solution. So that's where the support vector ideas come from. So we can have this separation margin, okay? So this region, okay? And then we want to have support vector identified once you decide you want to use this as a separator. As I said, these are the data points close to the separation. You also want this margin, okay, to be as wider as possible. It's called a hyperplane if you have high dimensional data. So for this 2D data, you can have this one straight line as the separation. But if you have high dimensional data, you're considering a hyperplane to separate them, to cut them into two different sections. Okay, so support vectors are the nearest data points to the separation line. Well, you also have to maximize this boundary. So this involves nearest or minimized and also maximized. So you found a minimized distance, which is from the space separation line to the data points, and also maximize the boundary. So that's the solutions of SVM. So in order to compute the distance, we will need to have a formula to compute the distance from data points from vector to a straight line. Okay, because we want to compute, if we decide we use this straight line as our separator, the first thing we need to compute is the distance between all the data to this separation line, because we want to find the support vectors. So which data point from two different classes is the nearest to the straight line. So we need to have a formula to compute the distance between every single point to this straight line you proposed, because you don't know whether your straight line is a good solution or not. The first step is to compute the distance. to this straight line. So we need an easy way to compute this distance. So we are going to derive a simple linear operation-based formula to compute every single dot to the straight lines. So I will illustrate how this will be computed. At the end, it's dot product. Okay, you can see, okay, how I compute if you give me a straight line and give me a dot. How the easiest way to compute the distance from the straight line, from the point to the straight line. The answer is dot product at the end. Okay. You can have this geometric proof if you have time to read them. At the end, you will find, okay, the distance, which is the position here to this straight line, which is the red part, is a formula which we can derive. There are also different ways to do this vector projection proof. So I will give you an example which I show you how this formula will be constructed. Okay. This is my own version. So it's, you see, okay, it's at the end is dot product between vectors. Okay, so in order to define a straight line, for example, you know you have y, you have a, x, b. That's a general formula. And this is slope. That's the intercept, okay? So our example, which I'm making, is a real numerical example. So slope in our case is 5/3, and the intercept is 5. So my straight line is y. If I draw this, will be 3 here, 5 here. So this is my example, okay? You will generalize later, so if it doesn't matter A and B is what number, as long as you know it's a straight line you select to separate the data points. You can change A and B to whatever numbers, but the general formula is like this, right? So we will compute a distance using this as an example. We will see, okay, where if I have a data, see that, for example, in this position here. Okay, so this is called, let's call it big X, and it has a position at X and Y. Give a name, a random point. So the distance I want to compute is from this to here. Okay. So how can I have this x1, y1? So this particular point, a random point. What's the distance to this? The first thing I will first is transform this. So if I transform this into x, So this is the same as that, okay? You just move around, and this is also a representation or formula for this straight line. Then I will extract from this formula a vector. This vector will be 5 and -3, which is this and that. I will also need, so this C will be, say, 15. So that's from the straight line. So if you have a general version of this, whatever, you can get this and that by doing slightly transformation, okay? And so where is this vector? This vector will be 5. It's somewhere here. So this is a W. But if you extend this to extend it further, you will realize this is a 90 degree. So this is 5 minus 3. This is guaranteed because of the transformation of this. Okay, this is a very special point. Let's call it V. Very general, xy, because xy is on the straight. Xy is on the straight line here. So let's just call this V is xy. So very general. Now we are going to compute. We have everything here. I will further extend this straight line to this is defined because you have this straight line definition is given, and this will be able to compute, and you can extend this. So I will first project my data point to this straight line, so that my projection will be from here. So this will be my projection. Project this vector to this. So you know how to do the projection. It's dot product. Basically, you have your x bar here times my w here. That's a dot product, that's projection. If you divide this by normalized version, so this expression is the distance, the projection, which is how long this part is. It's from 0 to here. So you project your dot point to here. So this is this part. So once we have this length, in order to compute the distance, which is this, you just need to minus this part. So this distance minus this part will be the distance we need. Okay? So we need the length of this green part. So what's the green part? Green part, we are going to use this V, very special point, to compute. So essentially, it's V project onto W. So we can have V times W. V times W. And normalize this, you will get the distance of this. Okay, makes sense so far? We did two projections. We project this given point to this vector, and then we project this to this itself. The angle is 0, so this now we are using the W to times this, okay? So you will realize this part is this part. You have V, X, and Y times 5 and minus 3. This should be negative C. So this will be negative C. There's a little bit of jump, but if you know the formula, you will realize that's the case, because xy is on this straight line. So the distance of this green part is just like this. So we can put them together, and the distance from this point to this straight line is this point times this W minus C. Then divided by the norm. So this is a D we need to compute. So that's the formula at the end we will use for any given point. If we want to compute his distance to this straight line, we just times W minus C divided by this. So everything inside this W and the C has been already defined, given by this straight line definition. So this computation is very easy. Okay? So if you still have some trouble and maybe spend some time on this, maybe I'm not so sure whether this should be positive C or negative C, but this is minus 8. Yeah, maybe this should be positive C, but yeah, I will work it out later, but this is just an idea. So at the end, you will get the formula this, okay? The point is, this is very easy to compute. So even you have a lot of the data, you just have your data points collections as a matrix. You can do this. And W is from this definition, the line you want to use. You change the slope, you change the value here, you change the intercept, you change the values in W and C, but that's just a minor operation. Then you through the dot product, you get the distance of all the data points to this straight line. Then from there you will select the support vectors. Well, you see a lot of those mathematical expression and with the W transpose with this plus. So this is a definition of this straight line here, okay? You see the minimized, you see the maximized operations. So minimized means you found the data points close to this separation line. Maximize means you want to increase the margin as much as possible. So you just change your slope, change your B, and try to find this balance, find the smallest support vectors and maximize the margin between the separation. So that's the operation you have until you get the W definition. Okay, basically you search for this A, these two numbers in W until you get this maximized separation. You can also derive the formula at the end. This margin will be just 2 divided by the magnitude of W, as long as you normalize everything. So this will be, you minimize this, you get maximized. So you change the W, change the values here, you change the slope until you get this maximized margin. Well, the problem comes in is as not every single case you will have this perfect separation. Sometimes you have these blue points cross the boundary, jump into the red points group. Well, you still have this good support vector, but you have to take this as outlier and consider them as a error message or noise. You want to use some kind of a soft margin in order to get rid of those noise. For instance, this may be too close. If you decide only use this right line as a separation, you will have a very narrow margin, and your new data points coming in may be misclassified into one group. But if you ignore this noise, 1 point, and select this as a soft margin, and this may be more robust. Okay. So in order to introduce this, there are some of these mistakes will be allowed in the loss function. So when you search these combinations of these numbers, you give some tolerance to the mistakes. So create soft margin and to tolerant to those outliers or support vectors tend to be misclassified. So this hard margin, ideal situation, and soft margin, you tolerance those small mistakes and still using the second closed data points as your support vector. Okay, for the next separation, we will consider the kernel approach. So now it's the title of this is sort of called kernel. So this is a transformation. you can have so-called kernelized SVM, not using the information in the given dimension. So we are going to increase our dimension. So most of the time you consider, data are too complicated, we need to reduce the dimension using PCA, for example, to have a clear idea. So the kernel idea is to use a kernel function to transform your data. Oftentimes you will increase the dimensionality of your data. So why you are making things more complicated? Because in the high dimensional space, those unclassified data points may be easy to separate. One of the ideas is like this: if you have red blocks, which red squares have been circled around with the green circles, there's no straight line you can separate them. There's no way you can have a support vector and any single straight line can separate them clearly. Always you have many mistakes, misclassification. However, if you do the transformation, you do the kernel and you get this into 3D, and you can easily separate them by introduce this decision hyperplane or this very clear cut. Okay, so this is the idea here. This is another easy understand idea. So if you have, so this is example, I'll show you. Yeah, let's take a 5 minutes break, and I draw on this, and we come back. Okay, so this is a small example is to be used to represent a scenario which we will have a samples in the input sample space. Then we will do a kernel function transformation to move them into a kernel feature space, which is higher dimension than the original. In our case, is this position is 1, 1, the green one here, okay? And then this green one is -1, -1. Well, in this two corner here is position have 1, -1, and this is a -1, 1. So now if you decide I want to use a straight line to separate them, then you can try, you cut here, cut here. There's no way you can clear cut them, okay? You always have a mistake because in this separation you put 3 in the same class, which mistake by 1. So the idea of the kernel is to do a transformation. Now we have this x and y position. This is a 2D space, x, y, to define each point. But now we are going to introduce this using a kernel function to increase the dimension to make it into 3D, x, y, and z. Okay? So this z is not randomly generated. It's by transform the variable in the low dimensional space. So in this transformation, z will be x times y. Okay? So this is kind of idea of the kernel transformation. Kernel function. If you select this kernel function smart enough, so let's see, f x y. So you can write this projection from this position to x, y, and x times y. Through this function, you will move your data to a higher dimensional space in which this data becomes the green one becomes -1, -1, 1, okay? And this becomes, after transformation, this becomes 1, 1, 1. This becomes -1, -1, 1. And this becomes 1, -1, -1. And this -1, 1. Okay, in this 3D space, feature space, you can easily find a separation, okay, by cut through this plane which cross 0, and by looking at the positive and negative of this third variable, you separate them clearly. So that's the idea about the feature, and you have different popular transformation combinations. In this case, we use A times B, and there are other options, okay? This is another illustration also from 1D to 2D, and by binding this, then you can find this straight line to cut through and separate blue points from the red points. Before it wasn't possible, so there's no way you can use any straight line to cut to separate them. One of the functions we use a lot is called a Gaussian kernel. So this is a Gaussian function. So you have your data points. In this case, x is 2 variable, 2D, then 3D, so you need x, y. But anyway, so you need to find the distance between your data points to another center. And use this transformation, you will express this as how close you are to the center Z, okay? If you are close, you will be have a high transformed value. If you are away from the center, you will be low. Which? Question again?
So, sir, instead of dot product, what if we take the cross product? That will also give a separation for these two points, right? Like determinant of
This one here?
Right.
Yeah, you can use different kernel functions if you have an idea, and I don't want to use product between x, y. You have others. What you can.
What I was saying, instead of x dot y, if we take x cross y, cross product instead of dot product, so that will give a determinant like x y result in a determinant which is like an area which will give you negative value for those.
It's not a dot product. It's just a two-number multiplication.
Okay.
Yeah. But you can have a different ways if you decide I have a better kernel function, and you can use it for sure. Yeah. So let me play this quick.
Hey, wait. I need you to show me how to convert a Word doc to a PDF again.
Seriously?
Yeah, so this is illustration of some of this Gaussian kernel idea. So you found the center of this blue point, the Z, okay? Then you just compute the distance between all the points to that center Z. If it's close, then you have raised it up more by this Gaussian distribution. And after this transformation, then you can cut across the section. But this illustration just is upside down, but it's the same idea, okay?
Product of some phi.
So this idea has been used in not only in classification. You will had kernelized the PCA, okay? So basically you transform your data before you do PCA, okay? You do this kernel function and increase the dimension to something that's easy for you to separate your points. So this is like a whole group of kernel methods. Okay, here there are some carefully selected kernel function. So this K. If you read this SVM literature or textbook, you always found this kind of expression. So K is your kernel function. Well, you see this kind of expression illustrates a so-called kernel trick It's a kind of a trick means some shortcut way and to find a very smart, smart way to use this carefully select kernel function to achieve something like this, okay? Dot product between transformed dot product. So in many operations in linear algebra, we need a dot product. We need to do this A, B, okay? So this is the expression of dot product between A and B. So idea about this so-called kernel trick is, so if you need dot product between two vectors, especially we have x, y in the original space, then you want to have a kernel function through the k. Then you transform them and into a high dimensional space in where you will so this become, for example, x, y, z, etc., become high dimensional space. But in high dimensional space, you will also need to do a lot of dot products within there. See, okay, see, this is small a in the low dimensional space, and in the high dimensional space become big A. And you also have a small b a lot oftentimes, okay? And after this kernel, you get a big B, x, y, z, sometimes even higher. Okay, so this is all we expect to see. But remember, in your operation after transform, you want to do SVM. So because the kernelized SVM. In SVM, we will do a lot of dot product. So you will have a lot of A B operations. Is that true? So because you will remember all this PCA, all this find the nearest distance from the straight line to the points. It's all involved dot product between points. And there's not very useful to do dot product here because they are not separable. Well, you want to do a lot dot product in the transformed version. So this process is computationally intensive if you have a lot of those transformation, because you need to do this kernel transformation for all the points and then to do this dot product in the high dimensional. This is computationally intensive. People found a kernel trick, the method, to tell you if you select this kernel transformation function carefully, smart enough, you will have the relationship between k, x, y transformed version. So let's see this function. So this is a transformed version. That's these two high dimensional space. Well, this relationship tell you you don't need to do this transformation, then dot product in here. You can just do one step kernel. You get a dot product directly. So you don't have to do all this by routine, common, official way. You can just through this transformation to get it. So example we had to illustrate is like this x, y, okay? This x and y are two vectors, okay, in this x and y. Two points in this low dimensional. The function, the kernel function is x1 times x1, x1 times x2, okay, to expand this. So this is a transformation. So if you transform x using this function, you will get this. Okay, so because x was 1, 2, 3, then you have this result, because 1 will be times itself, and the 2 will be 1 and 2. So you expand this original vector into this. Original was 3D, and then 1, 2, 3, 4, 5, 6, 7, 8, 9, 9D. Okay, you transform this to this and transform y to this. The dot product between this transformed version, the dot, you get the results 1024. Okay. However, you don't need to do this very complicated transform and dot product between transformed version. You can just using this kernel, which x and y dot product itself. So this is very easy to compute. Dot product itself, then squared, and you get that exactly the same result. So you don't have to go through. Oh, by the way, this is not a. This should be a transformed f here, right? So sometimes I got confused. So this will be the transformed function to make this higher dimension. And your kernel for this transformed function is like a set. So you don't need to do the transformation, then to do the dot product. You can just use the kernel function to apply to dot product in the before transformation. If your transformation is computationally intensive, you don't have to do this. You can use just kernel function to reach to the same purpose. And that's a numeric example to achieve that. So you don't need to do this transformation. Okay? So then, anyway, so this is a kernel trick has been used in a lot of this kernel-based method to do this computation more efficiently. So this kernel PCA idea is if you don't have a PCA transformation, you directly found a straight line of the first PC, you will collapse your data into here, and then you get something like this, as a standard PCA. Well, if you do the kernel transformation, you transform your data first, then your PCA will do the dimensionality reduction, but also you keep the separation of those data. We will try a kernel function to transform your data. So that's a data, the kernel function we use to transform your data, then apply PCA. So if before you don't do the separation, then you will get something like this as a result. But if you did the kernel version of the PCA, you get the separation like this. Okay, we are done with the support vector machine and the kernel function, transformation of this mapping function. Next, we are going to discuss the cell-cell communication analysis, right? So first, we need to have, we don't really need it, but it is relevant to cell-cell communication. So if you want to have two different cell types to communicate to each other, the chance for them to communicate will be if you have one group of cell which is only 10%, another is 90%. Would that communication will be strong compared to 50% each? Okay, so this is the idea about heterogeneity. If you have, so say 100 cells in total, then if you have only cell type 1, say 5%, and 95% are cell type 2. So the communication between them cannot be very strong because the heterogeneity level is not enough. We need, the best is 50-50, so the communication is higher. So this idea is to link to the heterogeneity definition, okay? Or sometimes people tell the diversity. The idea is if you have 50-50, then the frequency product between this will be 0.25. Well, if you have 90% in one, another 1%, your product between these two frequencies will be only 0.09. So this number is be used to quantify heterozygosity, and then this is the highest heterozygosity level you can get if you have a two binary separation of your population. Okay. So oftentimes people call it 2PQ instead of PQ. P is the frequency of the black dots, and the Q is 1 minus black dots. People want to do the 2PQ because you can have this P and the Q multiplication squared, and then you have this geometric interpretation of what a PQ is. So this is the area between this off-diagonal position, but the P and the Q are two frequency of two different binary elements. So in general, this heterozygosity definition is 1 minus the frequency of P squared plus Q squared. For K equals 2, if you only have a black and white member in the population, then this is a 2PQ. So in single cell data analysis, we utilize some similar idea, or also the expression level. So it wasn't possible if you have a bulk RNA-seq, you don't have cell type. But here, because you have a cell type, you can start to do this cell-cell communication analysis. Okay, it wasn't possible. It's very important biologically, because you need to have a communication between cells to help the cell and experience or subject to development and tissue homeostasis. And they have to, information have to communicate across organs even. And in immune interactions, you have this chain reaction between immune cells and between immune cells and pathogens. There are different categories of this communication. So sometimes it's called paracrine, and this is between ligand receptor mediated. And sometimes you need a physical contact. Just can create this. You have a physical contaction. If you communicate through this bloodstream endocrine, then you have secreted factors and carried on by bloodstream and to the far location of different, could be different organs. You can have autocrine, which yourself produce ligand and receptor back to yourself. So we will focus on mostly in the paracrine, this particular manner of the communication. There are many different ways have been, computational tools have been developed. This is only part of it, okay? It's a very long list. But all the ideas is based on the computation of this called interaction score between ligand receptor pairs, between cell type. So all this, okay, basically is the same idea. So you need to have a cell type annotation. You need to know your sample has this cell type A, cell type B, and cell type C. Then you want to study, so we want to focus on cell type A and B relationship. So you pick two cell types. You compute average expression of genes within their cell type, okay? Then you will look at the database, the ligand receptor, so which gene encodes a ligand and which gene encodes this corresponding receptor of that ligand. So you have the gene name, and this gene has been paired based on the ligand receptor relationship. Then you will look at the gene which encodes for the ligand and find its expression level in one of the cell type under study. Our example will be cell type A and B. And then you will look at the gene encoded for receptor and its gene expression level in the cell type B. With these two numbers, we will do a product to compute its interaction score. If this number is high, then this ligand receptor pair will be considered significant. Then you can see, okay, cell type A and B are talking to each other through this particular ligand receptor pair. So this is one of the database examples. It's called the cell linker. You can search by species and also by gene name. You can also download their ligand receptor, the whole database. So that's a table. You can look. This will be the gene encoded for the ligand, okay, CDH2, okay, and also its corresponding receptor by itself. Okay, this is like an integrin. Then you have ligand FGB and the secreted and CDH5 on the membrane as a receptor. So you have this already validated or previous known ligand receptor pairs in the database. And certainly this is species specific. Some of these pairs are not in the mouse. Some of these are only in one of the species. So you have to have this So if you have a pair, okay, say ligand gene, and you look at the cell type A, it has an average expression is 3. Well, this ligand A's corresponding receptor, receptor 1, in the cell type B have an expression level 7. So you can make a product between this, and you see, okay, cell type A and B are communicate each other through ligand A and its receptor A1. And there's a interaction score, which 21 by multiplicating this. You can also do the opposite direction. See, okay, cell type B have ligand expression level is 3. Will receptor is expressed highly in cell type A, so that direction will be from a B type B to A. So you also, in this case, have a interaction score 21. Simple production of these two numbers. We normally don't consider this scenario. So cell type A express receptor, cell type B express receptor, but they don't express ligand for that receptor at all. So there's no interaction. All these numbers too small, there's no interaction. Or they both express same ligand, but there's no receptor expressed in any of these cell types, so no interaction. So we only do this cross cell type ligand receptor pair product between their average expression. That's the principle, right? That's all you basically you need to understand the principle. It's just product of these two average expression. So I'm using examples as a real data as an example here. So this is a skin wound tissue cell from this sample. The GSM from the sample ID from the geo database. You will see this as a 10x genomics data, and you have matrix, you have gene, you have a barcode, which is cells barcode indications. So we process the data, do the dimensionality reduction, do the t-SNE embedding, okay, it's 3D. There's some QC, so you get rid of some cells and remove some genes. You get this. As I said, for cell-cell communication analysis, you need to have annotation, cell type. So we classify our cells and do the annotation. There are fibroblasts, macrophages, endothelium cell, and stellar cells. Then we need to pick two cell types to analyze the communications. So that's stellar cells as a lipid storing cells in the pancreas and the livers. So I'm focused on one of these pairs. One is ligand, another receptor. Maybe it's the other way around. Let's see. So this is from the database, okay? Their direct interaction.
How do we differentiate paracrine and juxtacrine?
Direct and indirect.
Paracrine and juxtacrine.
Oh, I don't know. They may have this indication in the database, whether they are through direct physical contact or not. I never pay attention to this conjugate screen.
Apart from the expression of the ligand and the receptor,
Mm-hmm.
other than that, do they consider the downstream of those ligand receptor interaction in addition to the expression of the ligand and the receptor?
Yeah, no, most of the analysis don't consider this. Yeah. Because the next step will get involved into internal cell gene communications and network, gene regulatory network. But that's a good question. So that's, I think a lot of people are trying to do this whole system modeling to look at the communications, then pass on the information to the downstream through this gene regulatory network. Yeah. So let's focus on, for example, these two, okay? I'm looking at this particular example and look at stellar cell and endothelium cell. What I found is if I compute, I also consider this fibroblast. If I compute for this pair, ligand receptor pair, I can compute the score. Look like this score is highest. This is from an endothelium cell to stellar cell. Remember, this is not symmetric. You have one direction, but the opposite direction may not be really significant. But it looks like this is a significant interaction, okay, pathway. So I look at the expression level of this ligand in endothelium cell. So you see there's really a lot of gene, a lot of cells express this gene compared to other cell type. For the receptor, this is not very specific, but at least for endothelium cell itself will not express that receptor. So it's not autocurrent. It's definitely this produce the ligand and secrete into the intercellular space, and then stellar cell will express that receptor for this ligand upregulated. You can also use this bar plot to see highly expression of this ligand in endothelium cell and relatively high expression of its receptor in stellar cell. So we built a pipeline to detect in so this pipeline we try to see, okay, if you don't have very good database, can we detect those unknown interactions? So this pipeline is a little bit complicated, but I just very quickly go through each step. It involved in the gene regulatory network construction, interaction score computation in the middle here, and the neural network to do this embedding. You will get some idea very quickly. So we start with expression matrix, then we have to annotate it. See, we found two types of cells. One is this purple and another is green. Then this is the Fokker cell types we want to study. So we separate them. And for each of the sub-matrices, we can build a gene regulatory network. So look at the genes correlation between genes. So these two networks will be almost the same because this contains all the genes shared by cell type. The method we use is called PC regression. So one network, so that's Felix, your question is within the cell, what happened if you have a communication. So we try to incorporate that information here. We build a network first for each cell type. Then we build this matrix for ligand. So this gene I is ligand and the gene J is receptor, for example. So build a matrix to compute this interaction score and fill up this big matrix. And some place like this is from I and J, two gene pairs, but this may not have any record in the ligand receptor database. But if you have a very high score, we still keep that into the matrix. As a result, maybe we detect some unknown ligand receptor pairs. And this matrix also not symmetric, because I to J indicate the ligand I to J with JI means the different direction. Let me see. Yeah, so JI should be J as ligand, I as receptor, and IJ means I as ligand, J as a receptor. So this is not a symmetric matrix. Now we have three matrices. One network, two network for gene regulation, and then the third will be this correspondence or communication score matrix. So we put the three matrices together and through the neural network to project genes into a low dimensional space. So this genes contains two sets. One set is from cell type 2 and another set from cell type 1. Then we will look at the distance between ligand receptor pairs, okay, from gene I in cell type 1. So you see two different colors of dots, and each dot is a gene. Okay, we will look at this, for example, this gene's expression position to that gene. If they are ligand receptor pair and also very close in their embedding space, their indication there's very strong communications between this particular pair. Because the information in the communication score and network, everything have already been incorporated using the neural network to do the embedding. So the close distance between two genes means there's some similarity between them, or communicate through communications or through the push of the network. So those genes which close to each other, it's also the gene pairs from two different cell types. If they are close to each other, small enough, then we will see, okay, these are significant gene pairs. And then we will sometimes we will search the database. If we don't rely on the database, we will suggest those close gene pairs are the new detected between these samples. So we can build this. So those are the examples. Two cell types is from fibroblast to the dendritic cell, from real data downloaded. We can detect the interaction between CCL19 to CCR7 receptor as a pair. Also, you look at up and downstream of fibroblast, you know, those genes immediately upstream or downstream the CCL9, and then after receptor receive this information, it will be passed on the ID3 gene downstream. Also, this ligand receptor pairs not only work separately alone, they'll have two pairs, okay, CCLR12 and have a receptor CCR4. Also, there's another channel to communicate between two cell types. So this picture will be very informative to allow you to understand the downstream, upstream, this communication, and also the combinations of this crosstalk through different ligand receptor pairs. So if you have 3 different cell types, we will do pairwise annotation analysis first, and then you can incorporate all the information together. For example, here we do fibroblast to dendritic cell and get this, and you can also do T cell to dendritic cell, so you get the communication through here. Then you have to synthesize all these pictures on your mind. So if you have multiple cell types in the microenvironment, it will be very complicated. You have a large graph like here. So now we don't have a good approach afterwards. What we should do? How can you combine, for example, these 2 detected network communications to give you a good story? So it still rely on your interpretation of your biology yourself. So I'm showing you the combinations of this 1, 2, 3 matrices. It's here. So this is network from cell type A, this network from cell type B. So the matrix, they are the same size because they contain the same genes. And this S and its transposed matrix are the corresponding matrices. So all these 4 will be put together to give to the neural network. So that's where this part is about. So for this part is if you have 2 samples. So this is, for example, this is from a healthy control. You have cell type A and B. Well, you also have cell type A and B from the diseased samples, patient samples. And we can put all these matrices together to do the comparison, this 2 sample comparison. You will get results which the ligand receptor pair significant in one sample but not significant in another. So the change, the strength of change of the ligand receptor pairs cross samples. So this is a very powerful analysis. Not any other approach. We just show you the long list of these existing tools can do this. Okay, so that's our innovation. We can do this embedding with the 2 samples, all the pairs together, to detect major changes in the ligand receptor communication strengths across samples. This is computational intensive because to put this huge matrix, if you have 1,000 genes, that will become 4,000 by 4,000. If you have 2,000, it will see the 2,000. This will be 2,000. Then you will have 8,000 by 8,000 size of matrices. We also are doing this comparison communication between this spatial transcription data. So if you know, okay, this is a tumor, and this is a immune cell, which the boundary around it. So you can do this communication between these boundary cells. So if you found the ligand receptor pair and a very strong communication between these neighbor cells, but not between non-neighbor cells, that's a good indication that this particular ligand receptor interaction probably is real and necessary for the crosstalk between immune cell and the cancer cell. So this is one of the directions we can move to do this cell-cell communications. by incorporating spatial transcribable data information. Okay, so that's all for this Tuesday lecture, and see you Thursday. Any quick questions? We're okay? All right.